Excellent. Thanks a lot. So then we move on to the next speaker. That's Luke Campagnola from Institute MindScope Program. And the title is Synaptic Dynamics in the Mouse and Human Cortex.
Thanks, Gota. Many thanks to the event organizers for putting together this workshop. I'm really excited to tell you all today about a project that I've been working on for the last several years at the Allen Institute in the Synaptic Physiology group. So here's a very brief overview. In this project, we've been using multi-patch recordings in mouse and human brain slices to characterize the behavior of individual synaptic connections. The output of this project is a public database of synapse properties that includes connectivity, synaptic strength, kinetics, short-term plasticity, things like that, and with a fairly coarse level of cell type specificity. So I'll get back to the techniques and the data in a moment, but first I want to talk just a little bit more about this idea of cell types. So this is a major area of focus for the Allen Institute to understand the different types of neurons in the brain. We believe that understanding these cell types, like having a complete parts list for the brain, is a foundational step towards understanding the system as a whole. So ideally, we would classify these cells based on their functional roles, but we don't really have access to that information. So in practice, we think about cell types in terms of properties that we can measure, such as how wide or narrow their action potentials are, or which plant species their dendrites remind us of, or more recently, what mRNA transcripts are expressed in the cell. In our case, we are using transgenic animals in which fluorescent proteins are expressed in specific genetically defined subclasses of cells. So in this picture, I'm showing you a few of our favorite subclasses. Parvalbumin and somatostatin are the well-known subclasses of cortical inhibitory interneurons. SIM1 on the left here is a layer 5 pyramidal cell type that projects primarily out of the cortex, and TLX3 is a layer 5 pyramidal cell class that projects primarily within the cortex. And we have several other types similar to these in terms of their cell type resolution. In mice, we can transgenically label 2 or 3 of these subclasses at a time in a single animal. However, we also receive living human brain tissue that has been resected in the normal course of epilepsy surgeries. So in those cases, we don't have access to these transgenic tools, and we fall back on other measures of cell type, such as morphology. But what if we could pick, right? So what properties of cells would we measure that most closely correlate with their functional role in the circuit? Well, maybe it's no surprise that my first choice would be connectivity. I want to know which cell types are connected by synapses and what are the properties of those connections. Likewise, if I were building a network model, I would want to give extra attention to these properties because I think they're probably the most primary important information to include in that model. Unfortunately, synapses are pretty difficult to access experimentally. So as a modeler, you might have a hard time finding the information that you need. As an example of this, this is work by Yazan Bila and others recently published in Neuron. This is an extraordinary collection of data from across the literature that summarizes basically everything we know about the probability of connection between different cortical cell types. This was used to determine the network topology in the mouse V1 model that was introduced a couple of days ago. And right now it's the best we can possibly do given all of the biological evidence available in the literature. But although I have very high praise for this work, I think there's a lot to be desired of the data from which it was derived. So these values are pulled from a wide variety of different sources. We have different species, different cortical regions, often very different experimental conditions, and in many cases, even just predictions based on cellular morphology. And that's just for connectivity. So if you wanted to go deeper and talk about the strength of these synapses or their latency or kinetics or short-term plasticity, well, then the data become much more sparse. So this is the major motivation for the project that I'm telling you about today, to fill as much of this matrix as possible with data that are collected in a consistent way under standardized conditions and from a single brain region. Okay, but I said this is hard to do experimentally, so let's talk about the approach. We use a technique called patch clamp electrophysiology. This is the basic idea. In these experiments, we begin with a piece of living brain tissue, and we pick 2 cells that we're interested in. At this point, we have no idea if the cells are synaptically connected. So we attach glass microelectrodes to the cells, and that lets us stimulate one cell to fire a series of action potentials while we record from another cell, and we watch for these electrical blips here. These are called postsynaptic potentials or postsynaptic currents, depending on the experiment, and here I'm just calling them postsynaptic events. If we see these events, then it's evidence that these 2 cells are connected by a synapse, and we go on to interrogate the properties of the synapse. We can measure the strength and the shape of the events and the latency and so on. We stimulate the presynaptic cells with trains in order to induce short-term plasticity. And I'll talk a bit about this later on in the talk. The benefit of this approach is that it's by far the most sensitive tool that we have to measure the dynamic behavior of a living synapse. The drawback is that this type of experiment is pretty laborious and time-consuming, so we can't do very many of them. The patch electrodes here are pretty challenging to work with, and each individual synapse takes like 20 minutes or so to characterize in our experiments. So to speed things up a bit, we just add more electrodes. We call this multi-patch electrophysiology, and with 8 electrodes, we can characterize up to 56 synapses in parallel. In most cases, an experiment like this would only yield a few synapses. So then we run this experiment many, many times on many different animals and with different combinations of cell types. And we report on the statistics of synapse properties as they relate to those cell types. So for example, for connections from TLX3 onto somatostatin cells, we could tell you the probability of 2 cells being connected and how that relates to the distance between the cells. Or we can see the distribution of event amplitudes between those populations of cells. We can see the kinetic parameters and so on. Okay, so that's the basic idea. Let's talk about some of the limitations of this technique. And we touched on this a bit earlier in the workshop. We experimentalists generally don't like to do this. We prefer to present our data in the best possible light and let you figure out the limitations later on down the road on your own time. But I think it's important, especially for modelers, to understand how the data should and should not be interpreted. So first, these experiments are done on living dissected brain tissue. It's been sliced into sections that are about 1/3 of a millimeter thick. It's been incubated in a dish and bathed in artificial cerebrospinal fluid. So all of the things in this process can affect basically every property we measure, from connectivity through short-term plasticity. That said, we do go to great lengths to minimize these effects, and generally our results match pretty closely to similar experiments that have been done in vivo that don't suffer from all of these effects. Another issue is that we only record from a small number of synapses per slice. So we show this big picture at the end that captures the population statistics, but it lacks a lot of the local structure that can only be seen by looking at a single complete brain. The cell subclasses that we use, such as somatostatin and parvalbumin, we use these to organize our data, but they're likely composed of many true cell types. So for example, like somatostatin interneurons might actually turn out to be a group of several different cell types, or many cell types per layer. Finally, we're really far from getting a complete picture of the process here. Synapses have long-term plasticity and learning rules. They can be modulated by many different neurotransmitters, and we're generally only looking at short-range connections in a small region of cortex. So there's a lot left to uncover here. Okay, so I think I've finally covered all of the introductory material. let's look at some actual data. I'm not going to go into a whole lot of detail here. I really just want to skim through some examples to give you a sense of what's available in the data set. So this is an overview of the synaptic connections that we have found during the course of the project. In these matrices, what you can see is the vertical axes here indicate the presynaptic cell type, and the horizontal axes indicate the postsynaptic cell type. And here we're breaking cell types down first based on their layer, 2, 3, 4, 5, 6. And then within layers, we're breaking them down based on their transgenic types. So for example, if we look at layer 5 parvalbumin cells right here, and we follow this over to layer 5 SST cells, we can see that we've tested for 116 of these connections in our data set, and of those 116, only 5 were connected. So that's a connectivity of about 4%, and so we color this element purple. The colors become more gray toward the edges where the sampling is low, so that indicates our relative lack of confidence about the values in those matrix elements. Over here for human tissue, we've looked at layers 2 through 6, but the majority of the data is in layers 2 and 3. And for now, we're only classifying these cells based on their layer and whether they're excitatory or inhibitory. We are currently working on subdividing based on other criteria, especially the intrinsic electrophysiological features of the cells. In the mouse data, we have 3 inhibitory subclasses per layer, PV, SST, and VIP, and 1 or 2 excitatory types per layer. And in both cases, you can see that we've concentrated mostly on intralaminar connections. The cross-layer connections here are much more sparsely sampled. Okay, so now if I'm building a model of cortex, what exactly should I do with all of these results? How should I use this data to connect up my network model? It's not totally straightforward because connectivity is represented here as a simple proportion of connections found in our experiments, but in the brain, connectivity is really a function of the spatial relationship between cells. So there are a few ways that we can get at this from our data. The obvious one is just to simply group our cells by the cell type criteria that you're using in the model, and then directly measure the spatial distribution of connections from our data. So here's an example of that where we've separated cells into excitatory and inhibitory. And you can see that that connectivity falls off pretty smoothly out to about 250, 300 microns. But you could also break this down by layer or by transgenic subclass or by the intrinsic properties of the cells, really depending on how the cell types are defined in your model. So really the approach here is to open up the data set and query it in such a way that it gives you these profiles that are relevant for the model you're building. We've also identified gap junctions in our data. Obviously, the connectivity pattern here looks very different, but it follows what's been seen in the literature before, that inner neurons only form gap junctions with members of the same cell class. So we really only see connections following along these diagonals within the matrix. So all that's a lot about connectivity, but to be honest, connectivity is not really our strength here. We're only sparsely sampling a much larger network, and it's my hope that the electron microscopy projects that we heard about earlier will eventually supersede our data set for connectivity, at least. Where our data really shines is when we look more deeply at the physiology of the synapses. So for example, we can measure the latency of each synapse, the delay from the presynaptic spike until the onset of the postsynaptic event. This here is a simplified view of the same matrix we saw earlier, where we're still looking at excitatory types across layer, but the inhibitory types have collapsed across all of the layers. One thing that stands out here immediately is this row right here for parvalbumin inner neurons that is much lighter than the rest of the matrix. And that just says that the projections of parvalbumin neurons have a shorter latency than anything else in the matrix. Now, if I wanted to incorporate all of this information into a model, I could just take the average values out of this matrix Or I could dig a little bit deeper and I could pull out distributions of latency values for each synapse type. Likewise, another kinetic parameter that we measure is rise time. So that's from the onset of the PSP to the peak value. A quick glance at this matrix shows that somatostatin cells send the slowest events, whereas parvalbumin cells receive the fastest events. And we have several other measurements that I'm going to skip over here, but we also look at the decay time constant, the event amplitude under different conditions, and so on. Earlier, I mentioned short-term plasticity, where the idea is the strength of a synapse can change transiently in response to the pattern of inputs that it receives. This is a pretty complex phenomenon, but the most simple version is that in response to ongoing activity, synapses can either facilitate, that is, they become stronger, or they can depress, they become weaker, or some mixture of the two. So one way to think about this is that facilitating synapses integrate their inputs over time, whereas depressing synapses respond to the derivative of their input. Another perhaps complementary way is to imagine that in your network model, you've got your large complex network with ongoing activity, but where certain elements of the network can transiently turn on or off based on the recent history of their activity. So that is to say that this is one mechanism by which the network can dynamically reconfigure itself in response to activity. The data on short-term plasticity are somewhat more difficult to find in the literature, so we're particularly excited to get these results out of our lab and into your models. We probe SDP in a few different ways. First, by stimulating the synapse with trains of different frequencies in order to induce short-term plasticity. And then we test the state of the synapse after different delay periods in order to see how quickly it returns to baseline. We also stimulate with random interval trains, but that is a whole trove of data that nobody has dug into yet. So I'm showing on the right here a couple of matrices. The top shows short-term plasticity induction following a 50 Hertz spike train, where the red elements are facilitating and the blue are depressing. The bottom matrix shows the level of recovery after waiting 250 milliseconds. So we see a well-known result here that excitatory inputs onto somatostatin cells are almost entirely facilitating, but we also see a scattering of facilitating synapses elsewhere in the matrix. And there are some elements for which we see a great deal of variability in terms of seeing both depressing and facilitating synapses. On the left, I have a few specific examples of cells showing depression, facilitation, and a steady response over many repeated trials. So the dots here are the amplitudes of individual synaptic events. And the one thing I want you to notice is how stochastic these synapses are. The events vary in size over a very wide range from spike to spike. And we've already heard some arguments earlier in the workshop in favor of including stochasticity in our models. So I've touched on a few different dynamic properties of synapses that are broadly recognized as essential to network function, but I think also overlooked in most models. And I think if we're serious about biophysically realistic modeling, this is one area that's going to need quite a bit more attention in the future. But to repeat a question we heard earlier, how do we take all of this data and build an accurate model from it? It's tricky because there are a lot of idiosyncrasies to the data and to the experiment that you are not necessarily easy to infer as a modeler coming to the data. So one thing we're working on is a system by which we can kind of wrap all of these up into a set of parameters that are easier to model. A common approach is to just come up with a model of synaptic release and use some minimization algorithm to infer the model parameters that best fit the data. There are a couple of things that I don't really like about this approach, and one is that these models can be very difficult to optimize, so you're never really sure if you found the best solution or just a local minimum. And another is that the experimental data that we produce is very rarely sufficient to constrain the models. Rather, there are very large regions of the parameter space that fit the data more or less equally well. So to deal with this, what we're basically doing is just brute forcing the parameter space. We optimize the fitting algorithm to the point that we can quickly test every point in a large parameter space. So that first, we're pretty confident about the optimal solution that we found. And second, we can report on the entire region of the parameter space that fits the data rather than a single solution. So in the plots above,
10 minutes, Luke.
Thank you. We are seeing slices of this parameter space with two different synapses represented. We have a PV to PV synapse in red and a pyramidal to SST synapse in blue. And this basically just confirms our expectations that the two synapses occupy very different regions of that parameter space. So again, the intent here is just that the output of this model search will be the easiest way to incorporate synaptic dynamics and stochasticity into your own models. But that's still work in progress. So I'm going to wrap up in the last few slides by talking about how you can get access to this data set if you're interested. We have a page up on the Allen Institute web portal that's got documentation on what we've done and how to access the data. You can find the address at the top of the slide here and for the next few slides. And I think these presentations will be available for download later on. You're also welcome to just contact us directly. We do have a second data release on a manuscript coming out later this year that will contain a lot more data and analysis, so stay tuned for that. If you do want to be notified when that happens, there's a link on the site here that you can request that from. We have a Python API that you can use to access the data. We have desktop analysis tools that we use to explore the data. And we have a whole bunch of Jupyter notebooks up that give examples of different analyses that you can do, including almost everything that you saw in this presentation. The data set comes in the form of SQLite database files. We encourage you to access these via our API, but you can also just download the database files and query them directly with your favorite tools. And that's it. I want to give a big thanks to many, many people across the Institute and outside the Institute as well. Projects like these are really difficult to do in a small environment. And I also want to remember Paul Allen, who gave us this opportunity to work in such an amazing and inspiring environment. So thanks for your attention.
Thanks a lot, Luke. Great talk, and there's a lot of questions, so I'll start reading them, and if you answer them quickly, we have time for more. So one has to do with like the possibility for high throughput measurements, and then there's Osman Sharifi is mentioning that, and Kwu Nok Mimi Man is asking, how about using optical activity sensor to increase the efficiency to find connected cells?
That's a great idea, and it's something that we have looked into a bit. The main limitation that's preventing that working right now is the amplitude of events that we're looking at in, especially in mice, are typically on the range of a couple hundred microvolts, whereas the sensitivity for the best voltage indicators we have is still off by at least a factor of 10. So the hope is that will arrive eventually, and we've already been waiting for that for many decades, but we're still not quite there.
And there's one question from Alexander Dimitrov. Do you see any correlations in neuron connectivity? That is, if A is likely connected to B, then A will also be likely connected to C.
That's a great question. Higher order connectivity is on our long list of analyses that have yet to be done on the data set. We unfortunately really haven't spent much time on that. However, the data set is public, and if you're looking for a project, we would love to have you dig in and tell us if you find any correlations.
So, and then there's one from Terry Sinofsky. What were the probabilities of release? And Ted has a follow-up, and did facilitation and depression manifest as changes of the probability for release?
What were the probabilities of release? That's a good question, and I actually have no idea. I went as far as running those models and visualizing them to make sure that they seem to be working. I actually haven't looked very carefully at the actual parameters that came out of that. And were they related to facilitation and depression? Absolutely. For example, let me see if I can find this slide again. On what are we looking at here? So we have on the y-axis base release probability, and on the x-axis number of release sites. And one of the things this is one of the ways that we can most strongly separate parvalbumin from SST synapses. Basically, anything that's facilitating is going to be in the bottom left triangle of this matrix, and anything that's depressing is going to be in the top right triangle. And these separate very strongly based on those parameters.
And then there's a question from Farsin. How can you be convinced that if one neuron responds to another one, responds to another one, then these two neurons have synapses in common? Maybe there's a third neuron or something else that make the relation between them.
Yeah, absolutely. That's another limitation that I should have mentioned earlier on. In the case of mouse, we're relatively confident that most of the data comes from monosynaptic connections. And one of the ways we argue that is just that we never see the situation where a PSP and ePSP coming in from one cell causes a spike in another cell. And another way we can look at that is just by looking at the distribution of latency values, where we believe these latencies are in a range where they're too short to be mediated by more than one synaptic connection. Now, in the case of the human data, that's not true at all. We've found many cases where stimulating a single neuron evokes polysynaptic chains that we then record in another neuron. So we're still kind of working through how we're going to separate the monosynaptic from polysynaptic, or how we're going to talk about that in our manuscript.
And there's one from Upi Bala. Have you been able to dissect the plasticity mechanisms a bit by running the experiments in the presence of selected pharmacological agents?
Yeah, there's an entire series of experiments that you can imagine applying pharmacology on top, applying different stimulation protocols, and we haven't been able to do any of that. So there's only so many experiments we can run, and our focus was on basically covering as much of that matrix as we could with these experiments, and going back and adding in different conditions to the experiments is something really interesting that we would love to do later on, but that's not something we have done yet.
And then there's one from, what happened to that one? Anyway, so something I'm interested in I'm wondering is, I don't know how, there's a lot of work doing this kind of paired patch recordings, obviously. But once you have the electrodes in, how much does it cost for you to run it longer so that you might be able to probe some of these like STDP rules and things like that? Because that would be, of course, a wonderful source for modelers.
Yeah, I think if I had to pick one thing that I could do to make this data set more rich, it would be to look into long-term plasticity mechanisms. But that's much more complex in terms of experimental protocols. And the real limitation in terms of the time that we spend on a cell is that after you attach this microelectrode to a cell, you've got at most an hour before the cell dies. But by the second half of that, you might even think, well, the cell's half dead anyway, so we're not really trusting what's happening here. So, we're more interested maybe in long-term plasticity mechanisms as studied without microelectrodes. And so looking at optogenetic stimulation, voltage imaging, things like that. But yeah, it's in the giant bucket of things we'd love to do.
Yeah, and thanks. Actually, thanks a lot for a great talk. You also got a special commendation from Alexander Dimitrov for sharing your limitations. That was appreciated. So thanks a lot.